Hello everyone. In the previous part we have discussed about the G alpha inhibitory subunits and the inhibition of adenylyl cyclase and about hyperpolarization, and in this part we have discussed about um, the transducin, that is G alpha T subunit rhodopsin here. So you can see here there is a rhodopsin, and this rhodopsin here light comes and this rhodopsin uh, detects the light. So here rhodopsin is a membrane protein. We all know that rhodopsin are found in rod cells. So here rhodopsin is a membrane protein that detects the light in the rod photoreceptor cell. So we have read so many G protein receptors. So here like other G protein coupled receptors, rhodopsin exists in equilibrium, and it has different forms like activated and inactivated. So this equilibrium is controlled by isomerization of retinol. Here you can see that 11 since retinol. So this retinol is the controller of this equilibrium, and the cofactor that is bound to the rhodopsin. So in the dark form, the retinal is in 11 cis form. In the dark, in the absence of light, the retinal that is present is in 11 cis form. You can see here, and it stabilizes rhodopsin, and it becomes inactive. The rhodopsin becomes inactive, and when light comes, you can see that is 11 cis retinol is converted into all trans retinol. You can see here when in the presence of light, and this retinol is photoisomerized to the all transform. 11 cis retinol is photoisomerized to the all transform, and then it catalyzes the replacement of GDP by GTP. Means this G alpha T subunits here attached to the GDP, and in the after activation of rhodopsin by the 11 cis retinol to all trans retinol form, in the presence of light, this GEP, that is guanine nucleotide exchange, GEF, sorry, that is guanine exchange nucleotide exchange factor, that converts that attaches the GTP and make the G alpha T subunit active. And after activation, we have already read that G alpha T leads to the activation of, after it becomes activated, it leads to the activation of phosphodiesterase, and the function of phosphodiesterase is conversion of CGMP to GMP, that is cyclic GMP to GMP. So here CGMP is responsible for the sodium ion channel activation. In the presence of CGMP, the sodium ions are in and out of the channel. So CGMP controls the sodium ion channel, but there is no CGMP formation. There CGMP is converted to GMP, that is no CGMP. So in sodium channel is also closed, and this G. So in the activation of this GTP attached G alpha T, here T is transducin protein, which is a type of heterotrimeric G protein, and it is a type alpha subunit which is bound to the rhodopsin and triggers the response of rod cells in the retina. So thus the mechanisms occurs, and